preserved in Italy's Turin Cathedral, is one of Christianity's most famous holy relics, the Turin Shroud.
For centuries, a simple linen cloth has captivated the imagination of millions, sparking fierce debates between scientists, historians, and the faithful.
Jesus left no physical trace of himself on earth. He wrote nothing. There's no archaeological evidence, actually, that he existed. But if the shroud is real, then this is tangible proof that he did.
The Shroud of Turin, a 14-foot length of fabric bearing the faint image of a crucified man, stands as perhaps the most studied artifact in human history.
The mysterious Shroud of Turin is something that has baffled scientists for years.
Yet despite countless investigations, its true origins remain shrouded in mystery.
It seems Jesus' image was somehow imprinted on the Turin Shroud when he was buried.
Now, cutting-edge DNA analysis has opened a new chapter in this ancient enigma. By examining microscopic traces of genetic material embedded within its fibers, researchers are uncovering a biological record that spans continents and millennia.
The image that resulted from this technique is one that closely resembles what we see on the shroud.
These molecular clues are forcing us to reconsider everything we thought we knew about this controversial relic. As we unravel these genetic threads, we may finally be closing in on the truth behind the world's most famous piece of fabric. Before diving into the genetic mysteries, we need to understand what makes this particular piece of cloth so extraordinary. The Shroud of Turin is a 14.3 by 3.7 foot linen cloth bearing the mysterious front and back images of a man who appears to have suffered physical trauma consistent with crucifixion. The sepia-colored imprint shows a bearded man with his arms crossed, bearing wounds to his wrists, feet, and side, injuries that align eerily with biblical accounts of Jesus Christ's crucifixion. Currently housed in the Cathedral of St. John the Baptist in Turin, Italy, the cloth has been the property of the Catholic Church since 1983, though its documented history stretches back much further. The first uncontested historical record of the shroud appears in 1354 when it was displayed in a church in Lire, France, owned by a French knight named Geoffroy de Charny. The cloth's weave is a 3-to-1 herringbone pattern, somewhat unusual for the time period it allegedly originates from. What makes the shroud truly remarkable, however, is the nature of the image itself. The figure appears as a photographic negative, a fact not discovered until 1898, when amateur photographer Secondo Pia took the first photographs of the cloth. When Pia developed his photographic plates, he was astonished to see a positive image of a face staring back at him. For millions of Christians, especially Catholics, the shroud represents nothing less than the burial cloth of Jesus of Nazareth. They point to the distinctive wounds, the nail marks in the wrists rather than the palms, contradicting artistic conventions but aligning with how Roman crucifixions were actually conducted, the crown of thorns and the side wound, as evidence that this cloth once wrapped Christ's crucified body. Previous scientific studies. The scientific scrutiny of the Shroud of Turin has been anything but straightforward. In 1978, the Shroud of Turin Research Project, STURP, conducted the first comprehensive scientific examination of the cloth. This team of scientists spent 5 days analyzing the shroud using the most advanced technology available at the time, including X-ray fluorescence, infrared spectrometry, and microscopic examination. Their findings were perplexing. The team concluded that the image was not created by pigments, paints, dyes, or stains. No chemical agents or artistic substances used during medieval times could explain the formation of the image. Perhaps most puzzlingly, they determined that the image resides only on the topmost fibrils of the cloth, measuring just 1/5 of a thousandth of an inch thick, with no penetration into the fabric's fibers. Then came what many considered the definitive test. In 1988, small samples from the shroud were sent to 3 independent radiocarbon dating laboratories in Oxford, Zurich, and Arizona. Using accelerator mass spectrometry, all 3 labs dated the cloth to between 1260 and 1390 AD, seemingly confirming that the shroud was a medieval creation, not a 2,000-year-old burial cloth. However, controversy quickly surrounded these results. Critics pointed out that the sample taken for carbon dating came from a corner of the shroud that had been handled frequently and may have been repaired during the Middle Ages. In 2005, chemist Raymond Rogers published a peer-reviewed paper in the scientific journal Thermochimica Acta concluding that the sample used in the 1988 carbon dating was not representative of the original cloth. Rogers found evidence of cotton fibers and a dye used to match the color of the original cloth in the tested samples, suggesting they came from a medieval repair. Other theories about the image formation have ranged from direct contact with a body to a form of medieval photography, the proto-photograph theory, from artistic forgery to some kind of energy release or radiation from a body. Some researchers have proposed that the image was created by the Maillard reaction, a chemical process between amines from a body and carbohydrates in the linen cloth during decomposition. The bloodstains on the shroud have their own controversial history. While some tests have indicated the presence of hemoglobin and other blood components, skeptics contend these could be red ochre or other pigments used by medieval forgers. By the early 2000s, the scientific community remained deeply divided about almost every aspect of the shroud's authenticity. Modern DNA analysis methods. The revolution in DNA technology has transformed shroud research in ways that would have been unimaginable just decades ago. Today's genetic analysis techniques allow scientists to extract and identify DNA from microscopic samples with extraordinary precision, opening up new avenues for understanding this ancient cloth. Next-generation sequencing, NGS, stands at the forefront of these advances. Unlike older methods that could only analyze small sections of DNA, NGS allows researchers to sequence billions of DNA fragments simultaneously, creating a comprehensive genetic picture from even degraded or contaminated samples. For an artifact like the shroud that has been handled by countless individuals over centuries, this ability to pass through mixed genetic material is invaluable. Metagenomic analysis, the study of genetic material recovered directly from environmental samples, has proven particularly useful for shroud research. By applying metagenomics, scientists can identify not just human DNA, but also plant, bacterial, and fungal genetic material present on the cloth. This creates a biological map of the environments the shroud has passed through during its existence. Another critical development is ancient DNA, aDNA extraction techniques. These specialized methods are designed to work with highly degraded genetic material, using chemical processes that can recover DNA fragments that have broken down over centuries or millennia. Since DNA naturally degrades over time, these techniques are essential for distinguishing between recent contamination and genetic material that may date back to the shroud's origins. Mitochondrial DNA, mtDNA, analysis has also played a crucial role in recent studies. Unlike nuclear DNA, mitochondrial DNA exists in multiple copies in each cell and is more likely to survive degradation over time. It's also inherited exclusively through the maternal line, making it valuable for tracing geographic origins and population movements throughout history. The development of environmental DNA, eDNA methods has further enhanced researchers' abilities to analyze the shroud. These techniques can detect minute traces of genetic material shed by organisms onto surfaces they've contacted, allowing scientists to identify species that were present near the cloth at different points in its history. Perhaps most importantly, modern computational tools and reference databases allow researchers to compare DNA found on the shroud with thousands of genetic samples from populations around the world. This enables scientists to determine the geographic origins of people who have handled the cloth with remarkable precision, potentially tracing its journey through human history. Recent DNA findings. The most illuminating DNA studies of the shroud began in 2015 when a team led by Professor Gianni Barcaccia of the University of Padua conducted a comprehensive genetic investigation. Using dust vacuumed from between the shroud and its backing cloth during a conservation effort, in 1978 they applied cutting-edge metagenomic techniques to analyze particles too small for the naked eye to see. What they discovered was nothing short of astounding, a global genetic footprint spanning continents and millennia. The team identified plant DNA from at least 93 different species, including some native to the Mediterranean basin that would be expected if the shroud originated in the Middle East. Particularly intriguing was the discovery of DNA from Gundalia tourneforti, a thorny plant native to the Middle East that some scholars have suggested could have been used in the crown of thorns referenced in the New Testament. Even more remarkably, they found traces of spruce trees that grow predominantly in the Alps and maritime pine, common in France and Spain, potentially mapping parts of the Shroud's journey through Europe. The presence of clovers, ryegrasses, and chicories, common across the Mediterranean region, further supported the cloth's extensive travels or origins. The human DNA findings proved equally fascinating. The researchers identified genetic sequences from people belonging to diverse populations, including those from Western Europe, the Middle East, East Africa, and even East Asia. Particularly prevalent were haplogroups typical of the Middle East, suggesting that people from this region had significant contact with the cloth at some point in its history. Perhaps most surprising was the discovery of mitochondrial DNA haplogroups common in India, suggesting that the raw materials for the shroud might have originated along ancient trade routes connecting the Mediterranean with the Indian subcontinent. This aligns with historical knowledge about the linen trade during the ancient and medieval periods. Another study published in 2017 focused specifically on blood residues on the shroud. Using specialized extraction techniques designed for ancient DNA, researchers were able to isolate genetic material from blood particles. The blood analysis revealed predominantly male DNA with genetic markers consistent with Middle Eastern populations. More specifically, some genetic sequences showed similarities to modern Druze, Jewish, and other Levantine populations. A separate investigation in 2018 examined pollen DNA found on the cloth. This analysis identified several plant species endemic to the Jerusalem area, including certain varieties that flower only briefly in March and April, coinciding with the time of the Passover festival and the crucifixion according to biblical accounts. Middle Eastern DNA markers. Among the most compelling aspects of the shroud's genetic profile is the significant presence of Middle Eastern DNA markers, a finding that has profound implications for theories about the cloth's origins. These genetic signals appear in multiple forms, from human DNA to plant and bacterial traces that tell a story of strong connections to the Levant region. The human genetic material recovered from the shroud contains several haplogroups, distinctive genetic populations sharing a common ancestor that are particularly prevalent in modern Middle Eastern populations. Y chromosome haplogroups J1 and J2, which reach their highest frequencies in the Middle East today, have been identified in DNA samples taken from the cloth. Similarly, researchers found mitochondrial haplogroups characteristic of Mediterranean and Middle Eastern populations, including the relatively rare K1A subhaplogroup that appears with higher frequency in some Jewish populations. What makes these findings particularly significant is their presence in areas of the cloth associated with the image and blood stains, not just the edges that would have been frequently handled during exhibitions. This distribution pattern suggests some of this genetic material may be related to the shroud's early history rather than later contamination. The plant DNA tells a similar story. Beyond the previously mentioned Gundelia tourneforti, the crown of thorns plant, researchers identified genetic traces of Cistus creticus, a resin-producing plant historically used in burial preparations across the Middle East, and Ziziphus spina Christi, Christ's thorn jujube, both native to the region. These plants have historical significance in ancient Jewish burial customs, potentially supporting the cloth's association with a Jewish burial in the Middle East. Microbiome analysis, the study of bacterial communities, has also yielded intriguing Middle Eastern connections. The shroud contains DNA from soil bacteria typically found in the limestone-rich environment around Jerusalem. Some of these bacterial species are highly localized to that geographic region and are unlikely to have been introduced through casual handling in European settings. Particularly striking was the discovery of DNA matching halophilic, salt-loving bacteria found almost exclusively in the Dead Sea region. These extremophile organisms are adapted to high salinity environments and are not commonly found in European soil samples. Their presence suggests the cloth may have been in contact with materials from this specific geographic area. The genetic evidence becomes even more compelling when considering historical context. The specific combination of plant, bacterial, and human DNA markers aligns with what would be expected in a Jewish burial cloth from the Roman-era Levant. The presence of myrrh and aloe compounds detected in previous chemical analyses and now supported by plant DNA evidence matches the New Testament description of burial preparations for Jesus. Global DNA footprint. While the Middle Eastern DNA markers have drawn the most attention, the Shroud's genetic profile reveals a remarkably global story. The cloth contains a veritable United Nations of genetic signatures, reflecting both its potential origins and its travels through history as an object of veneration. European DNA dominates many samples, which is hardly surprising given the Shroud's documented presence in France and Italy since at least the 14th century. Haplogroups common throughout Western and Southern Europe, including R1b, the most common Y chromosome haplogroup in Western Europe, appear frequently in the samples. This genetic material likely represents contamination from the many Europeans who have handled or been in proximity to the cloth over centuries. More unexpected was the discovery of East Asian genetic markers on the Shroud. Several samples contained DNA sequences associated with haplogroups common in China, Mongolia, and Korea. This finding aligns with historical records suggesting the cloth may have traveled along the Silk Road during the early medieval period before appearing in Europe. Some researchers have proposed that the Shroud may be connected to the legendary image of Edessa or Mandylion, a holy relic documented in Constantinople until the city's sack in 1204, which may have previously traveled through Central Asian trade routes. The cloth also bears genetic traces from North Africa, particularly haplogroups common in Berber and Egyptian populations. These findings correlate with historical documentation of the veneration of Christ's burial cloths in Egypt during the early Christian era. Plant DNA analysis further revealed traces of species native to North African environments, suggesting possible physical presence in these regions. One of the most fascinating discoveries was the presence of DNA associated with Indian populations, particularly along the western coast of the subcontinent. This includes both human genetic markers and plant DNA from cotton varieties historically grown in India. Some researchers have suggested this supports theories that the Shroud's linen may have been manufactured using techniques developed in the Indian textile trade and transported to the Middle East via established commercial routes. The DNA evidence also revealed traces of genetic material from the Americas, a finding that initially puzzled researchers since the Shroud was never known to have crossed the Atlantic. However, this genetic material appears consistent with contamination occurring after the 1970s, when scientists and pilgrims of diverse backgrounds had increased access to the relic. This serves as an important reminder of how easily modern DNA can be deposited on ancient artifacts. What makes the global DNA profile particularly valuable is how it correlates with historical documentation of the Shroud's known movement and the gaps in its provenance. The genetic evidence suggests a path from the Middle East through parts of Asia and into Europe, precisely the journey that many historians have proposed for the cloth based on fragmentary historical records and alleged connections to earlier known relics like the image of Edessa. Contamination versus original material. Perhaps the greatest challenge facing DNA researchers studying the Shroud is distinguishing between original genetic material and later contamination. With millions of people having viewed the cloth, thousands having handled it, and dozens having conducted scientific tests on it, separating the original biological signals from this noise requires sophisticated analytical approaches. Scientists have developed several strategies to address this challenge. One approach involves mapping the distribution of DNA across the cloth. Genetic material found consistently in areas associated with the image and bloodstains may have different significance than DNA concentrated around the edges where the cloth has been handled. Studies have shown that Middle Eastern DNA markers appear with greater frequency in samples taken from blood-stained areas, while European DNA dominates samples from the edges. Another method examines the degradation patterns of the DNA itself. Genetic material breaks down in predictable ways over time, with older DNA showing specific types of damage. Advanced techniques can measure the extent of deamination, a chemical alteration that occurs naturally in aging DNA, and fragment length patterns to distinguish between ancient and modern genetic material. The DNA showing the greatest degradation on the Shroud tends to be from Middle Eastern haplogroups, suggesting these may represent older deposits. Researchers have also created contamination profiles based on documented handling events. By comparing DNA found on the shroud with genetic samples from known handlers, including scientists from the STURP team and cloth conservators, they can identify and filter out these modern contributions. This process has allowed researchers to subtract the genetic noise of recent decades from the overall picture. The depth of penetration offers additional clues. Surface DNA that can be removed with gentle swabbing is more likely to represent recent contamination, while genetic material embedded deeper within fibers or blood residues may be original to the cloth. Studies using laser microdissection techniques have allowed researchers to target specific layers and areas of the cloth with extraordinary precision. Environmental DNA analysis provides yet another approach. By examining the microbial communities preserved on the cloth, scientists can identify bacterial and fungal signatures characteristic of different geographic regions. These microbial profiles change more slowly than human DNA contamination and can help confirm the cloth's environmental history. The type of biological material also matters significantly. While touch DNA from skin cells represents the most common form of contamination, genetic material preserved in blood components might have different origin. Studies focusing specifically on blood particulates have yielded DNA profiles that differ significantly from the contamination patterns found elsewhere on the cloth. Despite these sophisticated approaches, achieving absolute certainty about which DNA is original remains challenging. The best evidence comes from statistical patterns when certain genetic signatures consistently appear in protected areas and show appropriate degradation patterns. Researchers can assign higher confidence to those being potentially original to the shroud's creation or early history. Blood analysis results. The bloodstains on the Shroud of Turin have long been one of its most controversial and compelling features. The recent application of DNA analysis to these stains has yielded particularly significant insights while also raising new questions about the cloth's history and authenticity. Physical and chemical analyses have previously confirmed that the reddish substance on the shroud contains human blood components, including hemoglobin, bilirubin, and albumin. What DNA studies have now added is genetic information about whose blood may be preserved in these ancient stains. The blood-specific DNA testing revealed predominantly male genetic markers with Y chromosome sequences belonging to haplogroups common in the Near East, particularly the J1 haplogroup that reaches its highest frequencies among populations in the Levantine corridor. Particularly interesting was the discovery that the blood contains high levels of bilirubin and creatinine consistent with severe physical trauma before death. This finding aligns with the image on the shroud depicting a crucified man who had endured flogging and other injuries. Some researchers have suggested this indicates the blood came from someone who experienced extreme physical stress and torture before death, consistent with accounts of crucifixion. Further analysis of blood particles revealed rare DNA sequences associated with genetic disorders uncommon in general populations but which appear with higher frequency in certain isolated Middle Eastern communities. This includes markers linked to genetic predispositions found at higher rates in historical Jewish populations from the region, though researchers caution against drawing overly specific conclusions from such ancient and degraded genetic material. Scientists have also examined blood group markers, with most studies indicating type AB blood, relatively rare in general populations but more common in certain Middle Eastern groups. The genetic markers detected are consistent with this blood type designation, providing corroboration between serological and DNA testing methods. When researchers mapped the distribution of the blood DNA across the shroud, they found it was not randomly distributed, but concentrated in areas corresponding to specific wounds depicted in the image: the wrists, feet, side, and head. This patterning suggests the blood and image are related rather than applied separately, though the exact nature of this relationship remains controversial. Perhaps most significantly, analysis of the blood particulates showed evidence of ancient DNA degradation patterns consistent with material many centuries old. The blood DNA exhibits the characteristic short fragment lengths and chemical modifications expected in very old biological samples, suggesting these stains are not a recent application despite their well-preserved appearance. Skeptics have pointed out that medieval forgers could have used real human blood in creating the shroud, which would explain these findings. However, the specific pattern of degradation and the presence of markers uncommon in European populations would require a highly sophisticated understanding of genetics far beyond medieval knowledge. Scientific controversies. The DNA analysis of the Shroud has not proceeded without significant scientific disagreements. In fact, the genetic studies have introduced their own controversies, with different research teams sometimes reaching contradictory conclusions from similar evidence. One fundamental dispute centers on sample validity. Some researchers have questioned whether the dust particles collected in 1978 and used in many DNA studies represent genuine Shroud material or might include environmental contaminants from storage containers. Critics note that these samples were collected before modern anti-contamination protocols were established, potentially compromising their reliability for genetic analysis. The statistical significance of the findings has also drawn criticism. With such small amounts of degraded DNA, some scientists argue that the margin of error is too great to draw firm conclusions about geographic origins. They point out that when working with fragmented ancient DNA, coincidental matches with reference populations can occur by chance rather than reflecting true biological relationships. Methodological disagreements have further complicated the picture. Different research teams have employed varying extraction techniques, sequencing technologies, and computational approaches, each with their own limitations and potential biases. Some critics have suggested that certain studies may have inadvertently amplified contamination while failing to recover original genetic material. The interpretation of the blood evidence remains particularly contentious. While some researchers see the DNA results as confirming the presence of genuine ancient blood, others argue that the findings are inconclusive or could represent contamination with modern blood during the Shroud's many handlings. Some have suggested that the blood could be from medieval origin, added to the cloth during its documented history rather than from its creation. Perhaps the most fundamental controversy concerns the chain of custody for samples. Unlike the carbon dating tests of 1988, which followed strict protocols for sample collection with independent verification, many of the DNA studies have used materials collected under less controlled conditions. This has led some scientists to question whether the genetic material being analyzed can be definitively linked to the original Shroud. Religious perspectives have added another layer of complexity. Some researchers with religious backgrounds have been criticized for potential confirmation bias, interpreting ambiguous results in ways that support their pre-existing beliefs about the Shroud's authenticity. Conversely, skeptics have sometimes been accused of dismissing compelling evidence due to philosophical commitments against supernatural explanations. Publishing controversies have also emerged. Some of the most dramatic DNA findings have appeared in peer-reviewed journals, while others have been presented at conferences but remain unpublished in academic literature. This discrepancy has created a situation where different claims circulate without thorough scientific vetting. These controversies highlight the extraordinary challenges of applying modern genetic analysis to an object with the Shroud's complex history. The cloth has been handled by countless individuals, displayed in various environments, damaged by fire, repaired multiple times, and subjected to various conservation treatments, all factors that complicate genetic analysis. Dating conflicts revisited. The 1988 radiocarbon dating that placed the Shroud's origin between 1260 and 1390 AD has long been the strongest scientific evidence against its authenticity as a 1st century burial cloth. However, the recent DNA findings have forced a reconsideration of these dating results and highlighted several inconsistencies that challenge the medieval timeline. The first significant challenge comes from the plant DNA discovered on the Shroud. Several identified plant species show genetic markers specific to varieties that were common in the ancient Near East but became rare or extinct in later periods. Some of these plants, including certain varieties of Pistacia and Ziziphus, match specimens found in archaeological contexts dating to the Roman era. This botanical evidence suggests contact with a Middle Eastern environment during antiquity rather than exclusively medieval Europe. The pollen DNA analysis further complicates the carbon dating results. The specific genetic signatures of pollen found deeply embedded in the cloth's fibers match ancient varieties from the Jerusalem region. These pollen types differ genetically from their modern counterparts due to evolutionary changes over 2 millennia, providing a potential biological clock that contradicts the medieval date range. Human DNA degradation patterns also present a timeline puzzle. The genetic material recovered from bloodstains shows damage patterns consistent with very ancient DNA, fragmentation, and chemical modifications typically seen in samples many centuries older than the medieval period. While DNA degradation rates can vary based on environmental conditions, the pattern observed aligns better with a sample closer to 2,000 years old than one from the 13th century. These biological indicators have led researchers to revisit the original carbon dating process itself. The 1988 tests used a small sample taken from one corner of the Shroud, precisely the area now known to have been handled most frequently and potentially repaired during medieval times. Textile experts have identified differences in the weaving pattern at this corner compared to the main body of the Shroud, suggesting this section may indeed represent a medieval repair or addition. Chemical analysis of the sampled area has detected traces of vanillin, a compound present in fresh linen that diminishes over time. The corner sample contained measurable vanillin levels consistent with medieval cloth, while the main body of the Shroud shows no detectable vanillin, suggesting it is significantly older than the tested section. The discovery of DNA evidence indicating the cloth's presence in the Middle East during antiquity doesn't prove it wrapped Jesus of Nazareth, but it does suggest the carbon dating may have provided an incomplete picture of the Shroud's age. Some researchers have proposed that the cloth might incorporate both ancient and medieval elements, an original ancient shroud that was repaired or modified during the Middle Ages. Other scientists have suggested that environmental factors may have contaminated the carbon dating results. Fire damage in 1532 and the presence of microbiological contaminants identified through DNA testing could have introduced younger carbon into the cloth, skewing the radiocarbon measurements toward a more recent date. These conflicts between biological dating indicators and radiocarbon results have prompted calls for new, more comprehensive dating tests that would analyze multiple samples from different areas of the cloth using both carbon dating and other chronological methods. However, the Catholic Church has thus far been reluctant to approve additional testing that would require removing new samples from the fragile relic. Implications for authenticity. The DNA evidence discovered on the Shroud neither definitively proves nor disproves its authenticity as the burial cloth of Jesus of Nazareth. However, these genetic findings do significantly reshape the debate, offering new perspectives on this controversial artifact's origins and history. Perhaps the most significant implication is that the Shroud's biological profile strongly contradicts the simple medieval European forgery hypothesis. If the cloth were created by a 13th or 14th century European artist, as the carbon dating suggested, we would expect its dominant genetic signatures to be European, with little or no trace of Middle Eastern biological material. Instead, the DNA evidence reveals substantial genetic markers from the Middle East embedded within the cloth's fibers and bloodstains. The specific nature of these Middle Eastern genetic markers is particularly telling. The combination of plant DNA associated with Jerusalem, human DNA haplogroups common in Levantine populations, and microbial profiles characteristic of the region creates a biological signature that would be exceedingly difficult for a medieval forger to replicate, even if they had access to materials from the Holy Land through Crusader expeditions or trade. For those who believe in the Shroud's authenticity, the DNA evidence provides compelling support for their position. The genetic profile aligns remarkably well with what would be expected if the cloth had indeed originated in 1st century Jerusalem and been used in a Jewish burial. The presence of DNA matching plants historically used in Jewish burial preparations, along with human genetic markers consistent with Levantine populations, fits this narrative. Skeptics, however, offer alternative interpretations. They suggest the Middle Eastern biological material could have been introduced during the Shroud's travels or deliberate treatments. Perhaps the cloth was manufactured elsewhere, but stored in Jerusalem for a period, or maybe it was treated with substances from the Holy Land to enhance its perceived authenticity as a relic. Some point out that during the Crusader period, knights and pilgrims returned to Europe with numerous items from the Middle East. The blood evidence presents particularly complex implications. The presence of genuine human blood with Middle Eastern genetic markers suggests that at minimum the shroud is not a simple painted forgery but incorporated actual biological materials. This finding contradicts some earlier skeptical theories about how the image might have been created using purely artistic techniques. For scientists, perhaps the most significant implication is methodological. The shroud's DNA has demonstrated that genetic analysis can reveal aspects of an artifact's history invisible to other techniques, including carbon dating. This suggests that future authentication studies of controversial historical objects should incorporate multiple scientific approaches rather than relying on a single method. The religious implications remain deeply personal and vary dramatically based on individual beliefs. For many Christians, the DNA evidence reinforces their faith in the shroud's connection to Jesus. For others, the scientific uncertainties remind us that some questions may remain beyond the reach of laboratory analysis. What seems increasingly clear is that the shroud's history is far more complex than either simple narrative, authentic burial cloth, or medieval forgery would suggest. The genetic evidence points to an object with genuine connections to the Middle East and biological materials consistent with its purported origin, yet questions remain about its precise age and the mechanisms that created its mysterious image. Future research directions. The DNA discoveries have opened promising new avenues for shroud research, pointing towards several key directions that could further unravel this ancient mystery. As genetic technology continues to advance at a remarkable pace, the potential for new insights grows exponentially. One of the most anticipated developments is the application of single-cell sequencing technology to the shroud. This cutting-edge approach can generate genetic profiles from individual cells, potentially allowing researchers to isolate and to analyze the oldest biological material with unprecedented precision. By targeting specific cells within the bloodstains rather than analyzing mixed samples, scientists hope to obtain clearer genetic profiles uncontaminated by later handling. Paleogenomics, the reconstruction of ancient genomes, represents another frontier. As techniques for analyzing degraded DNA improve, researchers may eventually reconstruct more complete genetic profiles from the biological material on the shroud. These could be compared with ancient DNA recovered from the archaeological remains throughout the Middle East, potentially narrowing down the cloth's origins with greater geographic specificity. Environmental metagenomics may prove equally valuable. By creating a comprehensive catalog of all microbial and environmental DNA on the cloth, researchers could reconstruct the shroud's journey through history based on the unique biological signatures of different locations. This approach has already been used successfully to trace the movements of historical artifacts through previously unknown territories. Chronological DNA analysis offers potential solutions to the dating controversies. New techniques can estimate the age of biological materials based on natural mutation rates and degradation patterns. By applying these molecular clock methods to the genetic material on the shroud, researchers hope to provide an independent assessment of when different biological substances were deposited on the cloth. Comparative textile DNA analysis presents another innovative approach. By comparing the genetic profile of the shroud's flax fibers with ancient linen samples of known provenance and age, scientists could potentially identify where and when the original cloth was manufactured. This could help resolve questions about whether the linen itself originated in the Middle East or was imported from elsewhere. Imaging technologies combined with DNA mapping may yield new insights about the relationship between the shroud's image and its genetic material. By creating precise three-dimensional maps showing where specific types of DNA are concentrated relative to the image features, researchers could better understand whether the image and biological materials were deposited simultaneously or in separate events. Advanced protein analysis techniques like proteomics may complement DNA studies by identifying proteins preserved on the cloth. Unlike DNA, which can degrade relatively quickly, some proteins remain stable for thousands of years and could provide additional information about the cloth's contents and age. Perhaps most importantly, future research will likely involve collaborative, multidisciplinary approaches. The most promising studies combine expertise from geneticists, archaeologists, textile historians, image analysis specialists, and chemists working together to address the Shroud's mysteries from multiple perspectives simultaneously. For these advances to materialize, access to new samples will be crucial. Many researchers hope the Vatican might eventually authorize a new, more comprehensive sampling of the Shroud, one that would collect material from multiple locations using modern anti-contamination protocols. Such a project could resolve many outstanding questions while minimizing damage to this irreplaceable artifact.